Also take this opportunity to welcome you back to this other session of ours. Now before we begin, allow me clear this question out. There's somebody who said that master, please, for the questions in paper 2, those ones where they say compare, when you are comparing, somebody was confused that should I begin with the similarities or differences? The answer is you can begin with any. You can either begin with the similarities, you can either begin with what? Differences, as long as you write the word difference and you underline, or the word similarities and you do what? And underline. But to me, I prefer that child who has begun with the similarities, then followed by what? Followed by differences. So our topic today, we are going to look at the advantages. We are going to look at the advantages of C4 pathway, or the advantages of C4 plants. Now, when we look at advantages of C4 plants, one, we are seeing that C4 plants able to photosynthesize at very low carbon dioxide concentration, in bracket Ig, in dense tropical vegetation. This is because these plants have got what we call the PEP carboxylase enzyme, which has a very high affinity for what? For carbon dioxide. Then 2, we are seeing that these plants have what we call the concentric arrangement of the mesophyll cell, produces a small area in relation to volume for better utilization of available water and reduce the intensity of solar radiation. We looked at the concentric arrangement when we are looking at the leaf The leaf anatomy. Then we can also consider what we call photorespiration, which inhibits growth in C3 plants, is avoided, stock reduced in C4 plants because of the following. 1, the carbon dioxide fixing enzyme PEP carboxylase does not accept oxygen. Then 2, the rubisco enzyme inside the bundle sheath cells is shielded from high oxygen concentration by the ring of the palisade cells. Then the other point is that the carbon dioxide fixing enzyme in C4 plants are more active at hot temperatures and high illumination. So the PEP carboxylase is more active at hot temperatures and high illumination. Therefore, photosynthesis occurs rapidly at low altitude, hot and bright light, then tropical conditions than in C3 plants. Then the other one is that the productivity of C4 almost is 4 times greater than C3 because of the following. 1, we are saying that of the increased rate of carbon dioxide uptake caused by large internal leaf surface area. 2, we have short carbon dioxide diffusion distance. Then 3, we have carbon dioxide steep diffusion gradient. Then 2, we are saying that the bundle sheath cells in which the dark reaction occur have Roman number 1, a large photosynthetic surface area enabled by unusual large chloroplasts. Then 2, lack of the granular on which oxygen would be produced, so no photorespiration occurs. Then number 3, we are saying that the palisade cells in which light reactions occur have large granular to increase the photosynthetic surface area. So those have been the advantages of C4 plants over C3 plants. Then let us look at the disadvantages And normally, when we look at the disadvantages of C4 plants, it is only one, where we say that the carbon dioxide fixing enzyme in C4 plants are less active during cool, moist, and low illumination conditions. Therefore, photosynthesis occurs slowly at high altitude with the cool temperatures and in low light intensity of temperate conditions. NB, they are saying that C4 plants grow better under hot, dry conditions. When plants must close their stomata to conserve water with the stomata closed, carbon dioxide levels in the interior of the leaf falls and the oxygen levels rise. So that has been this session of disadvantages and advantages of C4 plants. I hope you have got that. So let's continue. Now we are going to look at the comparisons. Yeah, this is also very common. Comparisons. If you have time, you can open UNEB 2020. Is it 2022? Yeah, their number one paper 2 was about photosynthesis, autotrophic nutrition. By the way, that graph came from autotrophic nutrition. If you have time, go and read it through. Then let us look at the comparisons of C3 and the C4 plants. I said, yeah, when they talk about comparison, you normally begin with the similarities, then followed by what? Differences. So similarities, I said when you're writing similarities, use the word both, then the other one you can use then or while. It depends on how you are going to organize your work. So let us look at similarities. 1, both contain ribosco enzyme. I'm saying that both contain ribosco enzyme. Then the second point is that depend on light for their reactions. Then 3, both show carbon dioxide fixation. Then 4, in both they have the rubib. Then 5 form some organic products, e.g. pig, then PGA, sucrose. 6, they have the Kelvin cycle. Those are the similarities between C3 and C4 plants. Then the other one is the differences. As you can see there, the differences between C3 plants and the C4 plants. Now, when you look at the differences, so if you don't know, if you cannot separate them the way they have separated there, that structure and physiological, please don't. Just say differences, then you begin giving the differences of the two. Don't commit yourself to give the structure difference, then physiological, and you write that structure. You may end up making an error and you bring 1 point for physiological and you put it under what? Structure. So everything will be crossed out. Now here you can leave out if you cannot what? Separate the physiological and the structure. Now let's begin with the structure. We are saying that C3 plants, for them they lack the Kranz anatomy, while the C4, these C4 plants, for them they are having what we call the Kranz anatomy. I talked about this one. Then another point, all chloroplasts have identical structures in C3 plants. Then in C4 plants, we are seeing that chloroplasts are dimorphic. Dimorphic means in two forms. Di means two. Morphic shape forms. E.g., those of the palisade cells have grana, yet are lacking the bundle sheath cells. Then we look at the physiological. They're saying that carbon dioxide acceptor is a 5-carbon rubib in C3. Then C4, the carbon dioxide acceptor is a 3-carbon PEP, or is a 3-carbon compound called what? PEP. Then another one is saying that photorespiration occurs in C3 plants. Then we saw that in C4 plants we don't have what we call photorespiration. Then the other side, we have less photosynthetically efficient. C3 plants are less photosynthetically efficient. Then C4 plants are more photosynthetically efficient. Then C3 plant, we are saying that the GP is the first stable organic compound in C3. Then in C4, we saw that the OAA, or the oxaloacetic acid, is the first stable organic compound. We looked at this. Then the other one, enzymes are more efficient at low temperatures. Then enzymes are more efficient at high temperatures in C4 plants. Then the other side, rubisco enzyme is used. Then for this side, we have the PEP carboxylase enzyme is what is used. Then compensation point is attained at high carbon dioxide concentration. I will look at compensation point as we proceed. Then this side, compensation point is attained at low carbon dioxide concentration. So I will talk about compensation point as we proceed. I therefore want to end here, and I wish you the best. Please, I sign out.